I would like to welcome Dr. Stephanie Gomez from Children's National Medical Center, who's going to speak to us on product attributes of tumor-associated antigen T cells may inform clinical responses in pediatric patients with CNS tumors.
Thank you. First, I'd like to thank the organizers of this symposium for the opportunity to present this research. This is on our clinical trial evaluating tumor-associated antigen T cells in patients with brain tumors. So first, I'll give a little bit of overview of kind of the drug development process in clinical trials. So the success rate of drug development is only about 8%. So why is there such a large gap in clinical translation? Well, models are imperfect and don't always represent the human disease due to a variety of factors, and especially with pediatric diseases. They're very different biologically than adult tumors, so there's not as many models for the pediatric tumors. So I put this diagram up here to remind us all that the clinical trials are a really costly and lengthy process from the phase 1 trial to all the way through FDA approval. And a lot of trials do not go on from phase 1 to phase 2. And so what can we learn from early phase trials, like these early phase failures? Well, insights may lead to testable hypotheses and using experimental models. And this can lead to improved understanding of disease and mechanisms of therapy resistance. Insights also may directly lead to development of new therapies or modification of existing therapies. And therefore, we can address or overcome clinically relevant points of failure. So brain tumors are the leading cause of cancer deaths in children. CNS tumors are the most common and deadliest solid tumor in children. They often these patients undergo rigorous treatment plans. There's very toxic chemotherapy cycles, multiple rounds, also multiple rounds of radiotherapy, surgical resection. And in particular, diffuse intrinsic pontine glioma, or DIPG, is a highly aggressive brain tumor. DIPG is found on the brainstem and the pons, and it encompasses 10 to 20% of all pediatric tumors. There's no cure, and the overall survival rate is only 9 to 11 months. And so new and novel therapies are urgently needed to treat these children. So in this trial, we assessed whether adoptive T-cell therapy could be used for treating CNS tumors. And T-cells have some advantages. So T-cells can traverse the blood-brain barrier. They can specifically target tumor antigens and avoid healthy tissue. And these tumor-associated antigen T cells can recognize multiple targets to prevent immune escape. So the goal of this trial was to evaluate the safety of autologous T cells. So T cells that we took from the patient and engineered, T cells that we took from the patient and trained to recognize tumor-associated antigens, WT1, PRAME, and survivin. And then we infuse these back into the patients. So phase 1 clinical trials are not adequately powered to properly test efficacy of an experimental therapy, but we can look for some other things other than safety. We can look for signals of response or bioactivity. We can ask questions like, what is the best dose level? What is the best way to give the treatment? And how can we overcome potential barriers to efficacy? So this trial was a dose escalation trial. There are 3 dose levels, so dose level 1, dose level 2, and dose level 3 in each treatment group. And I'll be talking about 2 arms of this study. So arm A was comprised of patients that were newly diagnosed with DIPG, and these TAA T cells were given after completion of radiotherapy, which is standard of care. And then arm B is other recurrent or relapsed CNS malignancies, and these TAA T cells were given after completing the most recent course of therapy. So for the TAA T cell manufacture, PBMCs are peripheral blood mononuclear cells, were isolated from the patient's blood. And we pulsed them with peptides spanning the proteins WT1, PRAME, and survivin with antigen-presenting cells. And we did this over various stimulations. And at the end, we have our T cell product that we infuse back into the patient. So first, I'll talk about some of the patient outcomes in terms of safety. So most patients tolerated these infusions without toxicity. We're able to infuse the patients, and they can go home. A lot of these patients are able to go to school, and they feel, good enough to carry on with their lives. 2 potential infusion-related adverse events occurred, though. So patient 27 in Arm A, the DIPG arm, had a grade 5 adverse event. So this is potential pseudoprogression or tumor swelling 13 days after T cell infusion. And then patient 42 in Arm B had a grade 3 adverse event, so headache and potential pseudoprogression 23 days after T cell infusion. So next, I'll go over the outcomes in terms of survival. So this is for Arm A, the DIPG arm. We see here in the black dotted line, that is the median overall survival, historical median overall survival for this group of patients, which is about 10 months. So most of our patients are surviving longer than that. And in the green stars, so patient 13 and patient 22, we see a longer, an extended period of time for stable disease. So I looked to see what the products, these T cell products, what their characteristics were. So firstly, we did an ELISpot assay, so we assessed T cell reactivity to these antigens. So cells are one of the controls, actin is the peptide negative control, and then WT1, PRAME, and survivin are the peptides that the T cells should react to. And so this is measured by interferon gamma reactivity by looking at spots on this assay. So WT1, this patient 13 T cell product was specific for WT1, and we see a high, over 80% of TCR alpha beta T cells. So the TCR alpha beta T cells are the T cells that we would expect to be reactive to these peptides that would for the tumor associated antigen T cells. And the patient 22 had some reactivity to WT1, and PRAME was higher for that patient. And we also see a high TCR alpha beta population. So patient outcomes for Arm B, so the relapsed or refractory CNS tumors. This was encouraging too. We saw that these infusions may promote disease stabilization in these patients. So patient 9 had a very long period of stable disease, went off study, so we lost to follow-up. But patient 36 and patient 35 also prolonged period of stable disease. And I have the red star for patient 42, which was one of the adverse events that I spoke about. So when I look at the product characterization for these as well, these products that are specific by interferon gamma ELISpot for either WT1, PRAME, or survivin, and also have a high population of the TCR alpha beta T cells, seem to be associated with longer progression-free survival, or in the case of patient 42, potential pseudoprogression. And when we stratify the patients by dose level, so DL1 is dose level 1, DL2 is dose level 2, and DL3 is dose level 3. So when we stratify the patients based off of dose level, we see that the T cell infusions may prolong disease stabilization. So that's encouraging as well. So in conclusion, the T cells are well tolerated. They may prolong survival or promote disease stabilization for pediatric patients with CNS malignancies. And the T cell product attributes, so the phenotype and specificity, may inform clinical response. So future directions include clinical testing of personalized neoantigen-specific T cells in patients with CNS malignancies. This is our IMPACT trial. And I'm also going to be doing preclinical testing of TAA T cells in combination with DNA hypomethylating agents to upregulate the tumor-associated antigen expression in the tumor cells, and also upregulate antigen presentation so that the T cells can see the tumor. And since this clinical trial, there are a lot of people to acknowledge it's a team effort, and it's, takes a village. But I'd especially like to acknowledge my mentors, Dr. Kath Bullard and Dr. Jean Wang. Jean Wang is the brain tumor PI on the study.
Very nice. And again, it underscores, when you look at how long these patients are surviving, even the ones who did well, it's not good.
Yes.
So we have a long way to go. One of the questions that I had is, are patients eligible to be enrolled in these studies at diagnosis, or do they have to wait till they've been treated with something and then have progressive disease?
So for Arm A, it does have to be newly diagnosed, so basically right after completion of radiotherapy. So they do still get radiotherapy, and then they're enrolled on the study. And yeah, Arm B, they all have to have had previous lines of therapy. So,
Are there questions from anybody? I thought somebody's hand went. Yeah, Dr. Ladle.
Thanks. Yeah, I love these studies. Right, it's super, super exciting, super interesting. Do you have any? I mean, I guess you can characterize your T cell product going into it. I mean, how or what techniques are you using to kind of track those T cells once they're infused? Right, have kind of clonotypes you can follow, or can you see expansion of any of these products? And then, I'm sorry, I can't remember. Do you do lymphodepleting, like chemo or something, before infusions?
So for these two arms, we did not do lymphodepletive conditioning, but we have an arm C with, I think, 4 patients in that arm that we did lymphodepletion with. So we were seeing some encouraging results for that as well, but we're still in the process of doing more final analysis of that. And then in terms of your first, Track, yes. So for the TCR sequencing, we have done that a little bit. I am going back and analyzing again. It is hard to see these T cells in, the clonotypes in the peripheral blood. And my thought is that if they're doing what they're supposed to be doing, they're probably going to be mostly in the brain. But for future studies, definitely, we are going to be looking at that much more extensively. And yeah, so. Will be good.
Other questions? Yes.
Thanks. Great talk. Really exciting, and look forward to seeing where all this goes. Question I have is, so for CAR T cell therapies that have been trialed in DIPG, because of the sensitive location, right, and obviously with some of your adverse events, it's an issue, right? This just space, when you induce inflammation, swelling, it's a significant clinical problem. So I'm just curious if your all's experience has been similar to previously tried CAR T cell therapies, where these patients are essentially requiring active management of CSF pressure, active, pulling off CSF on a daily basis, sitting in the PICU with an open OMIA. Is that?
Yeah. So that's a great question. So we are giving steroids, so dexamethasone, when they do present, when they're hospitalized, due to this tumor swelling and brain swelling. And yeah, there are some more recent, studies with DIPG with CAR T cells for the GD2 CAR. It was Robbie Major and Crystal Meckel, and they relieve the CSF pressure. So we are, looking at different ways to manage that as well, and have an active collaboration with them as well. So yeah.
Thank you.